Hi, my name is Christine Byrne, and I've chosen to do prompt 2 for exam video 4, discussing the process of transcription in prokaryotes. So the purpose of transcription is to create a complementary RNA strand that's been synthesized from a single strand of DNA, which can later be used in the process of translation to create functioning active proteins. As a quick overview, I'm going to use an analogy of a scenario at a restaurant. I'm actually a waitress over at Gonza Tacos and Tequila on Hillsborough Street, and in the scenario, the owner has come in not to work, but to dine, and they will be representative of a DNA, while I, the waitress, will be representative of the enzyme RNA polymerase. So here it is. Hi Gonza, it's nice to serve you as the customer today. What can I get for you?
Hello Christine, it is nice to be served. Even though I know all of our delicious recipes, I'll leave that up to the chefs today. I would like to order the carne tacos with rice and charro beans.
Sure thing, I'll deliver the message to the kitchen. In short, the owner of the restaurant is similar to DNA in that both hold all the information necessary to create the desired product. In the scenario, tacos encells proteins. Neither, however, are the ones performing the actual work. That's where RNA polymerase, i.e. the waitress, comes in to transcribe or write down the order into a message, mRNA, that can then be delivered to the kitchen or to the rest of the cell. But let's get a bit more specific. DNA consists of two antiparallel strands of complementary nucleotides, meaning each strand runs in opposite directions and comprises a 5' and 3' end. This refers to the orientation of each nucleotide, specifically how the phosphate group of a single nucleotide attaches to the 5th carbon of its sugar ring, which then creates a phosphodiester bond with the hydroxyl group of another nucleotide that's attached to the 3rd carbon of its sugar ring. In the context of transcription, the DNA double helix consists of a coding strand and a template strand. The start site for transcription is at the +1 nucleotide downstream of the promoter region. This region contains a -10 tau box and -35 control region, two consensus sequences of the gene recognizable by a subunit of RNA polymerase known as sigma factor. It's this element that makes RNA polymerase a holoenzyme binding to the promoter region, breaking the hydrogen bonds between nucleotides, and creating a bubble within the double helix. Here we see that the template strand is the one with the free 3' end, allowing mRNA to be transcribed in the 5' to 3' direction. RNA polymerase then reads the template strand in order to join ribonucleoside triphosphates together, which will make up the mRNA strand. The sigma factor dissociates after about 9 to 12 RNTPs, allowing an elongation of the mRNA strand to continue with only the core RNA polymerase enzyme. This goes until termination, which occurs after an inverted repeat of nucleotides causing a hairpin loop formation signals termination, followed by a string of adenines. The adenine uracil bonds are then weak enough for dissociation of the DNA-RNA pairing. In a hypothetical situation, if the -10 tau box were to be deleted in a gene that codes for an enzyme to metabolize sugar, the RNA polymerase enzyme would be pretty lost trying to find the promoter region for which to bind. If RNA polymerase cannot bind to the DNA at the correct location for transcription to begin, it won't be able to correctly transcribe an mRNA strand that will later be translated into the enzyme needed for sugar metabolism. The consequences could be fatal for this organism if it relies on sugar as a necessary energy source.